Hello students, welcome back to VA NEET Academy. Hope everyone is doing great. Okay, so yesterday's upload was on Golgi Apparatus and today the class will be on lysosome, the next organelle. Okay, now how do you clean up your waste in your home? You might have seen your mom doing it, you might have seen your housekeep doing it, isn't it? So all the waste has to be disposed of, isn't it? So the similar function is done by our lysosome in the cell. So let's get started with today's class. Lysosome, the digestive system of the cell. Okay, let's see the definition. I'm sorry for my health today, so kindly bear. So definition of the lysosome. Lysosomes are membrane bound vesicles containing hydrolytic, or else you can say digestive, both are the same. Okay, hydrolytic or digestive enzymes. Okay, so because it's a digestive enzyme that digest, what does it digest? That digests the unwanted, okay, unwanted materials inside the cell. Okay, so that's the definition of a lysosome. Lysosomes are membrane bound vesicles containing hydrolytic, you can say hydrolytic enzymes or else digestive enzyme, both are the same. Certain books will say only hydrolytic, some will say only digestive, either way it's right. Okay, that digest unwanted material inside the cell. Clear? Okay, so let's talk about the discovery of lysosome. Okay, so lysosome, this was discovered by a Belgian scientist. Okay, Belgian scientist called as Christian Christian. Okay, Christian de Duve. Christian de Duve, D-U-V-E. Christian de Duve. I'll write it again. Christian, Christian de Duve, D-U-V-E, a Belgian scientist. Okay. So one of the NEET fact, or else most often asked in NEET, is that this is the same person who also coined the term lysosome. So he is the person who not only discovered lysosome but he also coined the term lysosome. Okay, hope it's clear, students. Now let's go into the structure of lysosome. Structure of lysosome. Structure of lysosome. So basically it has got a single membranous structure. It has got the single membrane structure. Okay, it has got digestive enzymes inside, and basically they are acidic interior. So from inside it is more acidic, or else you can say it has got low pH value. So that not a complicated structure, a very simple structure of a lysosome. You just have a vesicle, single membrane sac, you can say, and inside that there is lots of membranes, enzymes, hydrolytic enzymes. hydrolytic, or else you can say digestive enzyme. So, that is all the structure of your lysosome. So, nothing much. Yeah, so as I was saying, not a much complicated structure. You have a single membrane to be labeled, nothing much, and then these are, yeah, so we were talking about the lysosome structure, a single membrane structure, not much complicated. Okay, inside it we can find a hydrolytic enzymes. So that's the main function, main structure of a lysosome. Okay, so next see the formation of lysosome. How is the lysosome formed? Formation of lysosome. A formation of lysosome. So all of us know the enzymes are formed by ribosomes. Okay, ribosome, they attach together. Remember my class on ribosomes, the larger subunit, the smaller subunit, they come together in the presence of messenger RNA and convert the messages present in the messenger RNA into proteins. So enzymes are nothing but proteins. So these enzymes are formed in the ribosome. This ribosome is given to rough endoplasmic reticulum. Why is it given to rough endoplasmic reticulum? There it is being transported, modified, and then transported into Golgi apparatus. Okay, now what does the Golgi apparatus do? Golgi body do? It label it, it pack it, and then it is given into the, where? It is given into small sac like things. The packed material is given into a small sac like things, which you call it as lysosome, something like this. A small sacs pop out of the Golgi apparatus. Which side of Golgi apparatus do you remember? If you have seen my class, everyone should say it. It gets emitted or popped out from the trans end of the Golgi apparatus. Now let's talk about the functions of lysosome. Functions of lysosome. Okay, so one of the main important function of lysosome is intracellular digestion. Intracellular digestion. Intracellular digestion. Okay, so what do you mean by intracellular digestion? It digests the food, which is food particles, which is entering into the cell. So because intra means inside, cell means cellular. Okay, so it digests the food entering the cell. Second important function of lysosome is destruction of foreign bodies. Okay, for example, bacteria and pathogens. Bacteria and other pathogens. So it is responsible for the destruction of foreign bodies. Now how does this do? This uses the WBC cell. Remember, WBC is a white blood cell which use the lysosome to kill the bacteria and other pathogens. Okay, third function is recycling of cell components. Recycling of cell components. This is the third function of your lysosome. So how does it do that? It digests the old and damaged organelle. It digests the old and damaged organelles. Okay. So we were talking about the recycling of cell components as the third function. It removes the old and damaged organelles. Okay. So our own organelles, our own cells, they remove it, old, digest it, and this process is called autophagy. Okay, Now, how to remember autophagy term? Auto means self, isn't it? Auto means self. Okay, auto means self, our own. Okay, phagy is nothing but an eating process. So, eating the self. Okay, so it eats or else it digests the old and damaged organelles, and this process is called autophagy. So, once it is done, it is given back to the cell for the formation. Okay, so autophagy. Fourth one is autolysis function. Autolysis. So, what is autolysis? When a cell, okay, so when a cell is severely damaged, okay, supposingly a cell is there which is very badly damaged, okay, so lysosome, what it does, it releases its enzyme, into the cell, and this makes the entire cell to be eaten up by the hydrolytic enzymes of the cell, and this is what is called as autolysis. Auto means, as I told you, it is self. Lysis means breakdown, breakdown, whatever. Okay, so self breaking of the cell. So autolysis. Clear, students? Now this is this actually gives you a clue why is it called the suicidal bag, and which is a very favorite NEET based question. Why is lysosome called the suicidal bags? Okay, so that's because when the lysosome membrane, it ruptures, okay, it releases all the hydrolytic enzymes into the cell. Okay, so when the hydrolytic enzymes are released into the cell, it digests the cell itself. So this is why it is called the suicidal bag of the cell. A very famous and very favorite question of the NEET department or its NEET organization. So because the lysosome, when it ruptures, it releases all the hydrolytic enzymes into the into the cell, which makes the cell itself gets digested or else eaten up. Okay, so another question, important question, is where do you find lysosome in abundance? Where can we find lysosome in abundance? Okay, it is found in mostly WBC and phagocytic cells. Okay, it is found mostly in WBC and phagocytic cells, because these are the cells which tackle the foreign bodies of our system, foreign bodies entering into our system. Okay, so that's what that was about lysosome in today's class. Thank you, students.